Good morning, good afternoon, good evening, everyone, and thank you for joining us today. We are excited to have with us Kevin Corcoran, Vice President and General Manager of Agilent's Biomolecular Analysis Division, and Aaron Llanso, Associate Director of North America Market Development at Mission Bio. You may ask a question anytime throughout the webinar in the Q&A panel located on your screen, and they will be addressed at the end of the presentation. With that, Kevin, I will pass it over to you to begin today's presentation.
Great, thank you. Good morning and good afternoon, everyone. My name is Kevin Corcoran. I'm the VP and General Manager of the Biomolecular Analysis Division at Agilent. I'm really excited to be here today with our colleagues from Mission Bio, co-hosting the seminar on resolving tumor heterogeneity using single-cell technology and its importance in cancer research. Next slide. So here's the agenda for today, where in just a couple minutes, I'll turn this over to Aaron Llanso, who will detail the overview of the Mission Bio Tapestri Platform. And Aaron will share with you all the data examples of how the Tapestri Platform can be the next step for many of you on this journey to precision medicine. Back in April, I think this is the next slide, Aaron. Yeah, back in April, Agilent and Mission Bio entered into a co-marketing agreement in order to drive the adoption of the Mission Bio Tapestri platform. Tapestri is the only single-cell multi-omic platform that's capable of detecting both DNA and protein changes simultaneously, from the same cell. The Tapestri platform allows for the detection of clonal heterogeneity, which is extremely important in cancer. Agilent sees the partnership as complementary to our current portfolio, whereas we are the market leader in the creation of bulk targeted sequencing panels to detect genetic heterogeneity. Our technologies are really complementary in so many ways, including in the lab workflows. Since both technologies are NGS based, many of the steps, including the sample prep and the library prep, the sequencing and analysis are all very similar. And we understand how workflow is extremely important to our laboratory partners, specifically when approaching cancer research in a real multi-omics, multifaceted way, and looking for adoption of single-cell workflows to complement what is already part of your NGS lab. So I've mentioned cancer a few times during this introduction, but the truth is there are endless applications where Agilent's SureSelect technology and Mission Bio's Tapestri platform are complementary. We believe these complementary platforms can inform better decisions by our laboratory partners as we move more to this era of true personalized medicine. Using this multi-pronged approach, you can look deeper and be better informed of rare cancer phenotypes that may affect disease progression and therapy response. So with that, I'll now turn the seminar over to Aaron for a deeper dive on what the Tapestri can offer. Aaron?
Great, thank you so much, Kevin, and thanks for the introduction. And I wanted to add my welcome to everyone and thanks to the attendees for joining us today. I'm really excited to be here with our friends and partners at Agilent and with Kevin to share an overview of the Tapestri platform and some of the recent learnings and clinical utility that's come with the understanding of not only genetic heterogeneity through the use of powerful bulk platforms like the Agilent SureSelect platform and HaloPlex platform, but also moving that towards the clonal heterogeneity at the multi-parametric or multi-modal single-cell level. And so I wanted to start my talk today with this slide showing our mission statement at Mission Bio, because the first and most common question we get is, how are you different from other single-cell companies out there? There's quite a few of us, and it's a fast-moving field. So what distinguishes us in the space? And from the earlier days of the company, it used to be as simple as saying we're the only single-cell DNA sequencing platform to enable concurrent SNV and CNV genotyping from the same cell at scale. This is still true, but we've continued to evolve and to innovate to now enable multimodal single-cell assays off the backbone of the genotyping capability. And specifically, the latest edition of cell surface protein or co-detection with SNV and CNV with immunophenotype from the same cells. But beyond the technology itself, the real distinguishing answer is in our target applications. And we're largely focused on translational and clinical research, where we're enabling unique high-resolution analysis of clonal heterogeneity and disease evolution. There's really been an explosion of novel Tapestri data in the last year, and it's exciting to consider that with these advances in the field, that single-cell technologies may one day soon actually inform prognosis or therapeutic strategy in the clinic. So with that said, while most of the data I will share today is clinical research on primary samples, I do want to clarify up front that Tapestri is actually a research use only platform, an RUO platform. All right, so let's jump in. And I want to set the stage for understanding the methods behind the data that I'll share ahead with a quick overview of the Tapestri platform and discussion on why single-cell DNA sequencing is necessary. And this is really the next most common question we get is, why do we need single-cell DNA sequencing? Isn't bulk good enough? And what do I mean by that? There's lots of these new exquisite sensitivity applications have been demonstrated with ultra-deep error-corrected sequencing that folks like Todd Drewley and others have demonstrated in the field in studies of things like clonal hematopoiesis. And certainly with these newer techniques, and typically rare variants are being reported well below 1% VAFs in the research space, while clinical thresholds typically are still hovering in the 2 to 5% range. However, detection alone of a rare variant does not actually elucidate its clonal alignment. That is it a subclone or is it a distinct novel clone? And what somatic variants co-occur within this clone? And what are the cooperative implications of particular variant co-occurrence on clonal fitness? Are clonal architectures inferred from bulk sequencing? They're generally quite murky without cellular context, and in head-to-head studies, we've even shown that they can be incorrect. But that said, single-cell technologies really stand on the shoulders of bulk NGS giants before us, which have provided not only the sequencing backbone for single-cell assays, but they've also generated an incredible wealth of data over the last 10 years that have really helped to define the roles of genes and somatic variants that drive disease. And as sequencing costs have fallen, it's now increasingly more common to see a bulk sequencing study of a cohort proceed and importantly, critically inform the efficient design of a custom targeted panel for use on the Tapestri platform. And this is a nicely simplified cartoon that illustrates this point, whereas bulk DNA sequencing across the top here would identify a 40% variant allele fraction for each the blue and red variants in the tube pictured on the left. These VAFs are actually an averaged readout across the extracted nucleic acid from hundreds of thousands or millions of cells that are processed, thereby losing the cellular context of the mutational profiles. And it's essentially telling you the percent of cells that harbor that variant, but not telling you whether or not they co-occur and what that clonal architecture may look like. So in this example, the inferences from the bulk data would perhaps suggest a single clone containing both blue and red variants, or perhaps 2 single mutant clones. However, when we deploy a single cell sequencing approach, we can get direct and unambiguous assessment of the targeted mutation profiles within each cell, and can therefore identify 3 discrete clones, including 2 single mutant clones, 1 blue, 1 red, and 1 double mutant clone, each potentially affecting clonal fitness and having potential prognostic or therapeutic implications. And this is really the original problem statement for the Tapestri platform, which is rooted in the fact that cancer is a genetically heterogeneous disease, which has been demonstrated in multiple indications over and over. And we're deploying this approach to map tumor phylogeny, whereby continual accumulation of somatic mutations can lead to dysplasia or neoplastic disease with diverse clonal architectures that may yield unique implications for disease progression and prognosis. And this is really a key example of how the platform is being deployed today to sequence thousands of cells across longitudinal time points in order to track the effects of selective pressure of therapy on a tumor, and to understand what resistant clones are present or persist to drive relapse and refractory conditions within a patient, and learn how we may better inform therapeutic strategy moving forward. And as shown is in this example, what typically begins as a clonally heterogeneous disease can often collapse into a monolithic expansion of a refractory and what may have been previously a very minor or even undetectable clone that ultimately drives relapse in a patient previously deemed to be in remission. So for context, and before we get into data sets, I wanted to quickly share a high-level overview of the Tapestri platform, which consists of the Tapestri instrument, which is pictured there on the left. In the middle, we've got consumable reagent kits, which consist of the microfluidic cartridges, the ancillary panel reagents, NGS library prep reagents, and so on. And I'll go into a little bit more detail what that looks like in a minute. And it goes into the end-to-end bioinformatic workflow for which we provide great support and a whole host of software. That includes everything from custom panel design in our AI-powered Tapestri Designer, which is a free tool to support our customers in building custom targeted panels for both DNA and protein. And we actually just launched a brand new version, so I would encourage you to check that out at tapestrydesigner.com. And you can now design panels against the entire HG19 genome as well as the MM10 genome. And we've built in support for new gene editing approaches as well. In addition to the designer, once you've actually sequenced your samples, you can move into a cloud-based, or if needed, an on-premise variant calling Tapestri pipeline that does all of your genotyping. And then from there, you move into the Tapestri Insights, which is our visualization software for generating analysis and publication-ready figures. And then, of course, to wrap around all of that, we've got support from a highly experienced team of single-cell bioinformaticians, scientists that have broad experience in R and Python for custom tool development and are very nimble in the field to support our customer base. I also wanted to add and point out briefly that all of the software tools I just mentioned are actually provided at no additional charge when customers are purchasing our reagent kits. So on this slide, these are some of the frequently asked questions, and I wanted to provide just a list of platform specifications and guardrails for understanding the platform capabilities. As I've mentioned, Tapestri is the only single-cell platform capable of co-detection of single nucleotide variants, copy number variants, as well as immunophenotype cell surface proteins at scale. And to give you a sense, we're talking about panels, targeted DNA panels that can assay for hundreds of targeted loci, upwards of 1,000 amplicons at the higher end. And this can accomplish both gene level SNVs as well as chromosomal level CNV detection. And it's totally scalable from kilobase resolution on up to chromosome arm level detection as well. What do I mean by scale? It's scale, we're talking about thousands of cells from a single sample. So for this platform, it's currently 5,000 to 10,000 cells that you would expect to be processed and sequenced with genotype data provided at the end of the assay for 5,000 to 10,000 cells per sample. The sensitivity specification for the platform is down to a clonal frequency of 0.1%. We do have a handful of catalog panels, and there's quite a demand for customizable panels, which represents quite a percentage of our business at this point, and it's something that we're very well set up to support through the Tapestri designers I mentioned, and we've really built our business around being able to focus in on actionable or prognostic variants of interest within your project. And then finally, we do have the ability to work with solid tumor sample types. And we have a universal nuclei prep protocol that enables you to isolate nuclei from virtually any tissue type. We have quite a bit of experience at this point working with many different tissue samples. The one caveat right now is we are not supporting FFPE, but this is certainly of interest and on the roadmap, and we completely appreciate the importance of that sample type. It's just not something we currently support. So how does it work? In bulk, typically you're grinding up a sample, you're extracting nucleic acid, you're doing your sequencing, and you get an allele fraction for each detected variant within a sample. As I showed in the previous cartoon, you would not be able to resolve that into a clonal architecture. So how do we do that with Tapestri? So in the next line, Tapestri, as you see, we actually encapsulate each cell into a sub-nanoliter droplet, which actually serves as a tiny, discrete single-cell reaction vessel. And it's within this droplet that we're actually able to incorporate cell-identifying barcode oligo sequences into hotspot targeted amplicons that we amplify directly off of genomic DNA from the cell. These cell-identifying barcodes then enable us to bioinformatically deconvolve the bulk sequencing data and ascribe multiple variants to individual cells, thereby enabling us to resolve the clonal architecture as a function of the selected panel. Okay, so how do we do that? And another key differentiator for Mission Bio in the single cell space is we have a unique and novel two-step microfluidic workflow, which I'll walk you through now. And this is really key to enabling an efficient genotyping reaction through the use of a protease. And this is because protease digestion must occur separately from the downstream incorporation of molecular reagents into the droplet that would otherwise be destroyed in this reaction. So the first step is encapsulation, and that's what's pictured here on the slide, wherein a prepared cell suspension is loaded into the cartridge and co-flowed with a proprietary lysis and protease-containing reagent that then intersects a channel of encapsulation oil. So that forces these cells into the droplets. They're actually immediately lysed and encapsulated within this positive flow channel, which creates about 1 million droplets in 5 to 7 minutes. So this is a fairly quick part of the protocol. These droplets are then eluted off the cartridge into a strip tube, which is then placed onto a standard lab thermocycler in order to activate that protease. And this is a critical step that digests chromatin and nuclear-associated proteins that would otherwise inhibit efficient genomic PCR. Immediately following that incubation, the protease is heat inactivated on the thermal cycler, and the droplet population is then loaded back onto the second portion of the cartridge, and this is when we get into the barcoding reaction, pictured below. So in barcoding, the droplets then, and these are now containing our cell lysate that's been incubated with the protease, those droplets merge with a stream of hydrogel beads, which are the delivery vehicle for the cell-identifying oligo barcode sequences. So these beads are actually covered in a lawn of the same barcode sequence. And at the same time, PCR reagents and panel-specific oligos are added to the droplets. So now we've got all of our necessary reagents added for the downstream molecular biology for the genotyping piece to this assay. So once this step is complete, there's actually a UV light incorporated on the instrument, and there's a brief treatment that cleaves a photolinker sequence, which holds that barcode oligo to the bead. So that actually releases the barcodes into the droplet environment, where they're now subsequently available to be captured by a complementary sequence on the panel-specific oligos. So once these reagents have been added to the droplets, the sample is again eluted off the cartridge and placed back on the thermal cycler for 9 cycles of a highly multiplexed, now triple primer PCR. And that's essentially enabling us to look at the panel-specific loci. And while they're amplified, the cell barcode sequences are actually simultaneously incorporated into each amplicon. And that's important for us to be able to identify which amplicons are coming from which cells, from which drop. So now with the addition of the antibody oligo conjugates, I'm sorry, so I skipped one step. So after the amplification, and we've created an amplicon library, now that the barcodes are actually in the amplicon, we can actually break that droplet emulsion and go back into a bulk solution and clean up the reaction with spry beads and go straight into a Tapestri optimized Illumina-ready library preparation. And that's included as part of our kits. So now I've mentioned the immunophenotyping capability, which is not addressed in this particular workflow, but to address this, we actually just have a brief antibody staining protocol that's introduced up front of the first step. So when you have a prepared cell suspension, you're going to do a brief antibody stain and wash protocol. And with these antibodies, they're actually tagged with their own self-identifying barcodes. And after the staining and washing, the stained cells are then loaded, as I just described, onto the cartridge. And the protease will actually chew up that antibody, which leaves that oligo from the antibody behind, and that's now available to be captured into the library during the barcoding sequence. So these libraries are then separated for processing into separate Illumina libraries, one for DNA, one for protein, and then we can actually pull them back together for sequencing. So that's really the most recent adoption. The original protocol for the genotyping involves the encapsulation and barcoding, and then the antibody oligo conjugate, which enables the immunophenotyping piece, is just a brief addition protocol of staining and washing up front prior to loading the cells onto this platform. So another common question is, how does the single cell data compare to bulk data for variant calling? The short answer is, it looks highly concordant and recapitulates VAFs seen in bulk. And to look at this, as shown in this slide, we can actually run Tapestri and then bioinformatically discard the cell identifying barcodes that provide that cell context. And we analyze variant to low frequency in what we call pseudo bulk across the same sample. And as you can see from these 5 SNVs, we observe strong concordance with bulk variant detection. And in many cases, single cell can actually detect novel variants beneath the limit of detection of standard bulk assays. This is just to show that we are recapitulating the variants that are detected by the same bulk assay from the same sample. Okay, so now that we've covered the why and the how with the Tapestri platform, let's jump into some data, and I'm excited to share some use case scenarios from the last year or so. And I'll start in the hematological oncology space. We do have some examples in the solid tumor space that I'll move into as well, but I wanted to start off with a study that perhaps any of you that have attended a previous Mission Bio talk, this data will look familiar, as I would be remiss to not showcase the study from UPenn and UCSF, which was one of the first impactful stories to demonstrate how variant co-occurrence at the clonal level mediated resistance to a targeted therapy. And the one patient I'll dive into in the next slide demonstrates actually a particularly interesting presentation of how single cell may one day inform dynamic therapy approaches. So this study actually began in mid-2018. It was led by Dr. Sasha Pearl, Dr. Kathy Smith, and Martin Carroll. First author, Christine McMahon, on the publication that came out just over a year ago in May 2019. And this was a retrospective study of longitudinal bone marrow samples from a refractory relapse refractory AML patients treated on clinical trial protocol for Gilteritinib and monotherapy. Gilteritinib is a potent and selective FLT3 tyrosine kinase inhibitor. And they observed on trial that while many patients achieved a complete remission with this novel targeted therapy, many ultimately relapsed, and the investigators hypothesized, that this resistance, whether pre-existing or acquired, was mediated by the co-occurrence of an oncogenic RAS variants occurring together with FLT3 variants, and they wanted to confirm this. So the key questions here that they're posing is, when did the resistance develop, and what were the clones that were the dominant drivers of relapse in these patients for whom the FLT3 inhibitor therapy failed? And so, moving into this data, this is the typical variant allele frequency plot that you'll see longitudinally across multiple samples. And so this is one patient. These are actually pseudo-VAFs. So this is Tapestri data from which we've discarded barcodes to sort of illustrate this point. But they were able to provide 4 samples for this particular patient. The pre-treatment on Gilteritinib monotherapy. Obviously, these patients have been heavily pre-treated on prior lines of therapy. And then 2 on-treatment biopsy time points, and then the relapse time point. And what you see here is that there's a high allelic burden of FLT3. We see that IDH2 and SF3B1 practically overlap each other and are virtually the same VAFs. And then we see that NRAS, while it's either undetectable or very barely detectable in the earlier time points, really comes into become a main driver there in the relapse time frame with an explosive growth there. Now, what you can't say for certain is what the clonal architecture looks like, and that's exactly what we aim to resolve here and answer their hypothesis around the co-occurrence and whether or not that mediates the resistance to the FLT3 inhibitor. So we'll set that aside and dive into the single cell data. And when you actually run these 4 samples on Tapestri, we can now build this into a clonal architecture represented by colors here on the right in this bar graph. And so the cell numbers across the bottom show you the numbers of cells that were run for that particular sample at that time point. So at baseline, we had almost 14,000 cells run. And you can see that there's this very large triple mutant IDH2, SF3B1, and FLT3 containing clone that represents 87% of the tumor at that time point. We resolved that there actually is an IDH2 SF3B1 double mutant clone represented in green here. And then this dark charcoal color is actually wild type. And so we're not detecting what ultimately grows out to be one of the drivers of relapse in this patient, which is a quadruple mutant that appears to be this blue clone, IDH2, SF3B1, and FLT3, that has now acquired an NRAS mutation. And so what you see across this time course here is that the blue clone collapses and responds nicely to the FLT3 inhibition, and that's the triple mutant. And so while this is collapsing, however, you see that this quadruple mutant, which has acquired this novel NRAS mutation to co-occur with the FLT3 variant, is detectable as a very minor clone at the first on-treatment time point, as few as 3 cells at a 0.05% clonal frequency. But you see that start to aggressively expand. And so what was so interesting about this is not only did it confirm that there was a resistant population, a population of a clone that was resistant to the FLT3 inhibition in monotherapy, but we also see that there was another clone that was FLT3 negative. So it actually does not carry the target variant for that drug. And so as you might expect, that clone sort of continues to expand unchecked and is actually the dominant driver in this particular patient at the relapse time point. And so what was so interesting that really requires single cell technology here is to resolve it is that you see polyclonal drivers of relapse in this particular patient. So very powerful revelations in what happened in the disease course for this particular patient. Now, as I mentioned, and actually before I jump forward, I want to point out that, as I mentioned, there's a 0.05% clonal frequency. It's an extremely sensitive detection of this rare clone. And, as I mentioned before, in the specs, we have a sort of a specification of the system listed at 1%. However, that's largely considering for single mutant clones that may be quite rare, and it's difficult with sequencing error rates to determine, false positives. That said, when you have a genetically complex clone like this and you have multiple reads aligning to multiple variants in the same cell, even if that cell itself is a rare clone, in this case, we have 4 genetic pathogenic mutations that we're tracking. It actually increases significantly our confidence that this is a real clone when we start to see more cells. Now, retrospectively, we know that these variants were detectable by bulk, and so that also adds to the confidence that we can see here and speaks to the complementary nature of using bulk sequencing up front to profile a cohort of samples, build a targeted panel that's appropriate for that cohort, and move forward to resolve that into a clonal architecture and understand how the disease progresses. So with the understandings and learnings of this particular rare clone that we were able to detect, there was naturally an interest in minimal residual disease or measurable residual disease, which is a very hot topic, as I'm sure many of you are very acutely aware in the hematological oncology space. And so to that end, we partnered with a group at Stanford, the Ravi Majeti group, and they were particularly interested in understanding whether or not single cell sequencing can yield insight into measurable residual disease and whether or not that would be prognostic at the single cell level for patients that may go on to relapse or have refractory disease. And so they set up a study based on a few key points here, and that is that most patients that present with acute myeloid leukemia are able to achieve complete remission following induction chemotherapy, and that's typically defined by morphology being less than 5% blasts in the bone marrow. However, unfortunately, roughly 50% of patients are known to go on and relapse. And so Chris Hergen and many others have been looking at the implications of MRD and studying what it means if we're still able to detect residual disease in the clinical remission time point as defined by the morphology cutoff. So right now, MRD is currently measured by flow cytometry, digital droplet PCR for single variants, PCR, qPCR, so on. And essentially, what I want to highlight there is immunophenotype or immunophenotypic methods or genotypic methods, but separately in different workflows. And what's really been shown is that MRD, when detected at a 0.1% frequency, is associated with worse relapse-free survival and overall survival. So there's a really keen interest in characterizing which patients are in that risk category when they're in the remission time point. And it gives you the opportunity to potentially consider escalating therapy earlier before they actually clinically transform into a relapse scenario. But the beauty of the Tapestri platform is we can actually elucidate the clonal architecture genotypically together with an immunophenotype readout down to that 0.1% clonal frequency. And so this is of particular interest in simplifying workflows while hopefully aiming to improve the clonal readout for clinicians being able to make therapeutic decisions in the future based on this research. So in this particular study, they had 14 patients, 3 different time points. So the diagnostic time point, the clinical remission time point, and then at relapse. So this was done retrospectively, so we know that they all went on to relapse for the 10 patients that did ultimately relapse. And there were 4 non-relapse patients that were included for controls to understand what detection at that complete remission time point actually meant. So the key question here is really, can patterns of MRD and clonal evolution be detected with single cell genotyping? And this was genotyping only. So while I point out the immunophenotypic, that's sort of a forward-looking aim that I think will be very interesting to follow through on in future studies. For the purposes of this particular one, we were focused on genotyping. So what does the data look like? And I'll point you to the publication, which just came out in March of this year, which is fantastic. And I'll give you a few snippets of what some of these fish plots look like, as they're quite exciting to see this resolution of disease. So the first example I'll share is sort of a typical or maybe a classical one that you might perceive or think about relapse, where you have this monolithic clone that was treated successfully by chemotherapy, but we're actually in that remission time point able to detect as few as 6 cells of a quadruple mutant clone, this NPM1, PTPN11, IDH1, DNMT3A clone that ultimately is the monolithic driver of relapse in this particular patient. Again, these are the cell numbers across the bottom that were sequenced and genotyped for each sample time point. Another interesting one that is great when you're looking at clonal hematopoiesis, which is a known precursor to MDS and AML and other MPNs and such, is that when we can look at the clonal architectures, you can resolve what may be a clonal hematopoiesis clone that is not AML from overt AML clones that certainly need to be dealt with immediately, or if you're aware of them. And this fish plot demonstrates this nicely, where, again, we can detect a quadruple mutant, as few as 10 cells at that remission time point. You have a DNMT3A, IDH1, NPM1, NRAS clone that was the dominant driver both at diagnosis and relapse. And then, of course, this background that you see here is thought to be largely clonal hematopoiesis clones. So this may be a patient that is been substantially treated previously prior to this line of therapy, or perhaps is at advanced age, as we know that clonal hematopoiesis can correlate both with age and then prior genotoxic therapy. And then the third and final example I'll give here for this particular paper was an interesting presentation where you see this sort of heterogeneous presentation again of a dominant clone that's actually a double mutant NPM1 PTPN11 that is cleared nicely down to the remission time point. And then we see this clonal switching where there's just a novel clone that grows out to ultimately drive relapse. It becomes a double mutant NPM1 Wills tumor 1 at the remission time point that drives relapse in this particular patient. So getting this clonal level resolution enables you to understand, what are the implications at remission, and hopefully we'll be able to deploy that in the clinic in the future as valuable insight for clinicians when considering options for their patients. So they wrapped up this paper and provided a really nice outline of ways that they would conceive single-cell genotyping approaches being deployed in the clinic, as a means for MRD. And they really focused on 3 key cases where you would have CHIP that is detectable. So your clonal hematopoiesis clones of indeterminate potential. They're not disease necessarily, but they are precursors. So there's considerable, there's considerations for surveillance and risk there, but it's not overt disease at this point. So that can be also considered, together with MRD negativity, and you would potentially move that type of patient into a consolidation approach. If you have MRD positivity, then you've got residual disease there for sure. Then clinicians can consider escalating to allogeneic hematopoietic stem cell transplant. Or if they're MRD positive and there are targetable mutations, now you can consider whether or not there are other targeted drugs that may be appropriate for this patient, or if there are clinical trials for novel therapeutic agents that are being explored, you may be able to look at that at that time point for that particular patient. So these are sort of novel ways that they would see this sort of technology potentially being integrated in the future into the clinic in the hematological oncology space. And there's a lot of interest in that right now at multiple institutions. Importantly, they point out that clonal diversity is present in all baseline samples. So when they're running these initial presentation samples, you're seeing lots of pathogenic clones that are related to disease present, including both relapse and non-relapse cases. And then you see that clonal diversity decreases at remission, and that can correlate with relapse-free survival. Whereas if you do not see a decrease in clonal diversity, there's higher risk there and concern for that patient in terms of relapse-free survival and potentially overall survival. So I wanted to pivot a little bit from hematological malignancies over to the solid tumor world, where we've done some very interesting work with a number of folks and looking at clonal evolution of solid tumors. And one of the first ones that we were very fortunate to work with Dr. Charlie Swanton's group over at the Crick Institute in Europe. We worked on some previously characterized renal cell carcinoma samples to look for CNV events. And so this was work earlier last year where we wanted to flesh out the ability of the platform to actually call copy number variants. And the way that we do this is that we can design amplicons in a given loci of interest, and that may be a single gene, or if you have specific regions that are larger than genes that span multiple genes all the way up to chromosomal arms, we can tile amplicons across, and you're looking for generally a sweet spot of about 5 amplicons, across which we can then normalize numbers of reads for those amplicons and compare that to wild type clone populations that are either present in the sample, as they frequently are and very detectable even when you have high tumor burden, or we can actually spike in, CNV controls that enable us to calculate against wild type or the control and get to integer level copy number that enables us to do things like call loss of heterozygosity, copy gains, copy losses, and deletions. And so here, this is some brief data showing you the amplicon positions where we're seeing a loss of heterozygosity represented as a copy number of 1 for these amplicons positioned across chromosome 3, chromosome 9, and 14 in this renal cell carcinoma study. And this is something that we are excited about, because you can now look at the co-occurrence of single nucleotide variants together with copy number variants in a holistic genotype approach to understand how these tumors are evolving and what the drivers are, whether that be in a hematological tumor or a solid tumor. So we moved forward in some work with Dr. Swanton's group to look at some metastatic melanoma samples. And if you're familiar with the TRACER-X study that they're leading, very exciting capability that enables them to look at the clonal evolution of disease across multiple metastatic sites. And so this is a rapid autopsy protocol. And in this particular case, this was primary metastatic melanoma, where we see that there are 2 chest wall metastatic sites, a lung metastatic site. We ran some normal liver, excuse me, and then a MET from a different region of the liver. And these are some images just to show you, what that protocol kind of looks like, some of the H&E, and then, the fact that we can successfully isolate millions of nuclei from, small chunks of tissue or a couple slides of tissue. And again, these are cryopreserved or fresh frozen. We can dissociate cells and run them through, or you can do nuclei. This particular example of data is run off of the universal nuclei isolation protocol that worked quite well for getting into this tumor type. And so having run those samples with our tumor hotspot panel, you can actually look at the phylogeny of this particular tumor and infer, based on complexity of co-occurring variants, which clone may be the founder. And so in this particular case, it was inferred that there are 2 heterozygous positions in KDR and BRAF, the V600K variant, and the KDR is a non-coding SNV. That's inferred to be the founder clone in this particular tumor, and we map out how that evolves. And so the first clone that evolves from there acquired a new heterozygous position in the NRAS Q61L position. That became a homozygous variant in the secondary clone from that. And then the third clone acquired an additional heterozygous variant that's non-coding in the ImmTOR. On a different branch, we see that clone 4, as we're calling it here, actually has a homozygous acquired NRAS variant in G13R. And then the 5th and final clone that was detected acquired an additional heterozygous BRAF G593D mutation. And so what's cool about this is when you map that into phylogeny, you can understand the branched evolution of this particular tumor to understand how it evolved, and then we can actually overlay that in the body to understand which clones were responsible for colonizing which metastatic site in the body. And so as you can see here on the right, the diameter of the circle is representing the approximate clonal frequency of that particular clone, and the color corresponds with the phylogenetic tree as you see on the left here. So what's interesting is that in the lung MET, you see that it is the sole presentation of clone 5. So this BRAF G593D clone is only detectable in the lung site. And you see that precursor of the 14.1% lesser frequent or more minor clone of the NRAS G13R clone. There's lots of interesting studies looking at co-occurrence and how these clones evolve and how additional somatic hits may lead to gains in clonal fitness. And so, you see here that clone 4 is believed to have evolved from the inferred founder, but it's actually clone 5 that really took off and became that monolithic clone in the lung metastatic site. And then you can see the corresponding wild type here in the right-hand column. So 15.7% wild type, which we can easily distinguish from pathogenic clones. And then going on through the rest of the metastatic sites, you can see similar presentations between the 2 chest wall mets. However, one does have that ImmTOR clone that is not present or detectable in the other chest wall met. And then in the liver met, you've got a similar presentation where all 3 of these sites seem to have similar trajectories. They've got clone 1 and 2 present. It's just one of the chest walls that happens to have that ImmTOR clone, and then you see the corresponding wild types there on the right. So for time, I wanted to move quickly into gene editing, and this is another very exciting area of innovation in the single cell space. Gene editing is really coming up as a new frontier of potential therapeutic efficacy in a way that we've not seen for so many different diseases, and there's so many different applications. And of course, there's major considerations of the accuracy, reproducibility of gene editing approaches, of which there are many. And so being able to resolve the resulting clonal heterogeneity becomes an important question when you're thinking about incorporating this into a human treatment. And so the National Institute of Standard and Time, or NIST, has created a gene editing consortium, of which we have, we're proud to say we have joined, along with many other big names and partners in the field, to build out and convene experts across academia, industry, nonprofit, and government to really address the need for quality control or testing of these gene-edited products to understand and provide additional confidence around genome editing approaches and ensuring that when we're aiming to incorporate a fix, that we're not actually accidentally incorporating off-target edits or that perhaps only one of multiple edits was incorporated. And so we're involved in a number of early proof of principle studies here to characterize this capability. And again, I'll show you, I'll start off this data here with the bulk variant allele frequency. And this is a study with a partner here that is looking at 3 specific loci where they're aiming to incorporate homozygous edits. So it's actually a total of 6 edits. And from this variant allele frequency bulk sequencing data after the incorporated edits, you do see that all 3 have been successful to varying degrees. The first on-target edit, we're detecting a VAF of about 61%, and then that ranges down to the third on-target edit that is somewhere in the 12%. So you see different efficiencies. And of course, there's some questions and concerns about whether or not all 6 of these edits are actually occurring within the same cells. And when you see different VAFs like that, it's kind of hard to conclude that. Again, when we're looking at edits on both alleles, you're not getting zygosity information from the variant allele frequency data shown here in bulk. So if we set that aside and we move to the Tapestri data, you'll see that we actually were able to resolve quite literally every combination of edits that you would be able to see from this study with 6 different edit sites. And it was actually less than 1% successful where all 6 edits are completely incorporated into the intended clone. And you see different zygosity states or editing efficiencies across all of the other resulting clones, some with only 1 edit that occurred, and of course the different zygosity states with the light blue representing heterozygous edits, whereas the darker blue color is a homozygous edit. And so the point here is that there can be major clonal heterogeneity that results from these gene editing approaches. And so this is actually now being deployed by a number of our partners to optimize their editing approaches. And so this particular partner in some subsequent studies was able to significantly improve the editing efficiency so that all 6 targets were incorporated with a success rate of well over 50%, which is a massive improvement compared to the data shown here. And for time, I won't go into more of that. But so, we started off talking about the importance of multimodal single-cell sequencing, and that's what's really exciting and new from Mission Bio these days is, we launched with the backbone genotyping capability, which has been quite impactful in translational and clinical research from the outset. And now we're really moving to expand our capability. And as I've mentioned, the immunophenotypic capability is our first step in that direction. Many of you likely saw that the 2019 Nature Method of the Year was single-cell multimodal omics, if you will. And there's many of these types of combinations out there in the field today. And we're excited to be contributing the genotype and immunophenotype capability as our initial offering towards, multi-access or multi-parametric data from single cells. And so to illustrate the importance of multi-omics at the single cell level, this is a quick study we did in-house where you're merging 4 different cancer clones, some of which are quite similar to each other, and measuring them with different combinations of analytes here. And so if we take our sort of classic S&V only approach, you see that you've got the orange and yellow clone really clustering together on this t-SNE separation. They don't separate because the SNPs in these particular cell lines are virtually identical, and therefore we only see 3 clusters separating out. And those are your K562 and your TOM1 cells. They're very similar in clustering on SNPs. If you look at CNV, you see something similar, but it's different clones where you've got the blue clone and the yellow clone not being able to separate very well here. These are the Raji and the TOM1 cells that are quite similar from a copy number aberration perspective. Similarly, if you look at immunophenotype only, you sort of get even messier separation. But it's really when you start to merge these analytes together and separate across multiple analytes at the single cell level that you get much better separation. So if you look at copy number together with protein, you now see that we can mostly distinguish all 4 clusters here fairly well. But when you merge single nucleotide variants together with copy number variants and immunophenotype together, now we get very clean separation of all 4 clusters. And this really illustrates the power of multimodal analysis to resolve clones that may be similar or some that have evolved from other clones within to be able to isolate those and study them in the same workflow will require this type of approach. And so to enable this, we've announced late last year, a partnership with BioLegend, and they will be scaling up production of antibody oligoconjugates that are compatible with our platform. And so you'll be able to multiplex these AOCs, as we call them, the antibody oligoconjugates, together with our multiplex targeted amplicon panels to be able to look at genotype together with immunophenotype. And they will have both catalog and custom antibody panels available. And we expect that to launch a little later this year. Although currently we are supporting, as in the interim, the service capability to support our customers with antibody oligoconjugates in the meantime until this is up and running. So as a brief example, I'm really proud of the work that we've done with the Ross Levine group. They're a fantastic group of collaborators to work with. They recently put out a preprint on bioRxiv that I would point you to. Please don't rush to jot this down. You may have seen my Twitter handle for both Mission Bio and myself on the very first slide. And certainly please feel free to reach out to me directly, and I can point you to it or provide those links. We may also be providing these slides after the presentation. So this is one of the first papers that have come out on this platform using the novel genotype and immunophenotype capability. And this is work led by Lindy Miles together with Aaron Vinny and Bobby Bowman in the Levine lab. And so they started off their work initially by building a custom 109 amplicon DNA-only panel. And then they were the first to deploy a Mission Bio made antibody oligoconjugate panel to look at how clonal fitness of co-occurrence of DNA genotypes at different stages of differentiation or different cell types, as identified by immunophenotype, appear in many in multiple different hematological disease states, from clonal hematopoiesis all the way through to MPNs and AML. And so they processed, as part of the study, 146 primary samples from 123 patients, so one of the largest studies of its kind, certainly, and one of the first studies of this kind using this technology. And the aims of the project were really to understand the implications of how mutations co-occur and what that means, and what are the impacts of co-mutations on the immunophenotypic landscape of that particular clone, and is there heterogeneity within? And I'll just briefly, for time, summarize their results, and I would encourage you to review their data. And there are many presentations online of Ross sharing this data as well, and he's certainly far better than I to share the insights. But in brief, the numbers of mutations in clonal complexity was observed to increase, and this is probably expected. So from a sort of pre-cancer or precursor state of clonal hematopoiesis, where you're seeing a couple of epigenetic mutations occurring, leading to MPN, which now we have additional somatic hits, and then you're seeing the largest mutational burden in clonal complexity in overt AML patients. And they also observed that certain mutations and combinations of mutations effectively drove clonal expansion. There's a number of really interesting phylogenetic trees, similar to the one that I pointed out in Dr. Swanton's work, where you see that a secondary clone actually had significantly advanced clonal fitness beyond that of a precursor clone. And they are also seeing that. And it's very interesting to see how they map the trajectories of these clones based on the co-occurrence of different variants in different immunophenotypes. And these are frequently seen in the dominant clone of sort of recurrently combined mutations. And they were able to correlate significant differences in the immunophenotype, so the differing levels of surface protein expression across the different mutant clonotypes. So this is really interesting, ability to look at this in the first time. And Ross actually has a video, as I mentioned, on our YouTube as well, that you can review of their presentation. But just to give you a snapshot of what this looks like, this is a UMAP separation of different neighborhoods colored in by genotype. And so these are your clonotypes here. And so with this genotype distribution, you can see where the different clonal populations are. And then you can overlay or color in a heat map color spectrum here that shows you the expression level of a cell surface marker, such as CD45, for each neighborhood. And so you can start to see that you've got higher levels of expression of CD45RA in this particular neighborhood down here, represented by some of these blue variants, this DNMT3A Y874C clone. And then you can start, you see that there's a markedly reduced expression level CD45 in this particular cluster up here. And so you start to see that there is heterogeneity between the different clones that are present in each neighborhood, and similarly for CD11B and many other markers that they ran. And then if you play that out and look at, you can build heat maps off this and show correlations between co-occurrence of mutations and the effect on these immunophenotypic markers. So here for CD34, when you have a co-occurrence of DNMT3A and IDH1 variants, you see an increased expression of CD34 here, whereas conversely, if you see co-occurrence of IDH1 and DNMT3A, we see a significant reduction in CD11B production. So Just some brief snapshots. Again, I would encourage you to review this preprint. It's not yet published, but very exciting work and some of the first to use this approach. So with that, I want to go ahead and wrap and thank the many people that have been involved in all of the work that I shared with you today, all of our customers and collaborators from UCSF, Dr. Kathy Smith, Dr. Sasha Pearl, and Martin Carroll, and Christine McMahon from UPenn, Dr. Majetti and his team from Stanford, the fantastic group in Dr. Levine's group at Sloan Kettering, that's Lindy Miles, Ross, Bobby, and Aaron. And then all of my colleagues at Mission Bio, our funding sources, of which there have been many. And if you have additional questions, we'll certainly take them now. And please feel free to reach out to your Agilent representative if you have specific interest in a project, or if you have questions about applications or capabilities of the platform. You can also reach out to us at info@missionbio.com. So with that, thank you very much, and we'll take questions.
Thank you, Aaron, for that great presentation. We will now begin a Q&A portion of our webinar. You may put a question using the question and answers box located on your console. To ask a question over the phone line, please click the raise hand icon located at the bottom right corner of your WebEx window. I will then receive a notification and unmute your line so you can ask your question. Due to timing, some questions might only be answered offline. I will send over a list to the team as well as your contact information, and they will reach out directly. So one of the first questions, do you have a recommended extraction protocol to run upfront of Tapestri?
By extraction protocol, I'm guessing that we're referring to a disassociation protocol for disaggregating tissue into cells, because we're actually going to input cell suspensions into the platform, where then we're not actually doing a nucleic acid extraction. It's actually a crude lysate that's in the droplet itself. So I assume that it's a question about the dissociation if you're working with solid tumor. And for that, I will say that we don't have a specific protocol around dissociation at this point, but there are a number of kits commercially available out there. And I would recommend getting in touch with our support team to ensure that we review protocols for ones that you're familiar with. I think there are a couple that we may be able to point you to. Importantly, sometimes there are DNases in a couple of those kits out there. And of course, with a genotyping approach, we want to make sure that we're not going to accidentally digest all of the nucleic acid in the early step there.
Great, thanks, Aaron. I have an Agilent SureSelect or HaloPlex panel. Are they compatible with the Tapestri platform? And if not, how can I convert this and run on a Mission Bio Tapestri panel?
Yeah, that's a fantastic question. The short answer is yes, they are compatible. The longer answer is a little more complicated. With this being an Amplicon design panel in single cell, coverage can vary when you're considering various aspects of a crude lysate amplification at single cell resolution. So what we would do is actually take the content from your Agilent panel and put that into our designer software that I mentioned, which is actually very easy to do. And generally speaking, we would expect to see very good coverage of the same exact variants of interest, and we can work with you to iterate and make sure that any critical points or content within an Agilent panel is prioritized for high coverage in any Mission Bio panel. But yes, the short answer is yes, we can absolutely convert Agilent panels to Mission Bio Tapestri panels.
Great, thanks. Another question, what are the differences between Tapestri and 10x CNV analysis pipeline?
Yeah, another great question. So the CNV application from 10x, as I understand it, and I'll be the first to admit, I'm not an expert in the 10x workflows, but the 10x approach involves a whole genome copy number. So you're going to have much lower resolution CNVs that are detectable in a given sample. Because we're taking a targeted approach, you can focus in on regions of specific interest with much more sensitivity and much more granular resolution, down to the kilobase level, to look at individual genes of interest, whereas you're sort of hoping that you get enough read coverage from a whole genome approach to capture implicated copy number events with that kind of approach.
Great, thanks. What quality metrics does the system provide to ensure I'm only looking at the data from a single cell and not Droplet?
Yeah, that's another great question. We've got a lot of great bioinformatics engineers and specialists that have quite a bit of experience in the software side, building out things like artificial intelligence and machine learning and deep neural networks and things that are well beyond my understanding. And you know, they have been able to use that in a variety of ways that really have significantly improved things like, you know, Tapestri Panel Designer, which uses artificial intelligence, but also to inform quality control metrics and understanding, you know, whether or not a doublet or a multiplet is present, and that can be, you know, caused by 2 cells or multiple cells encapsulated into a same droplet, or if you have multiple barcodes. And there's a number of ways that you can detect that by looking at things like how we call a cell or panel uniformity. And typically you're going to see genotype qualities and numbers of reads fall off considerably when you have multiple cells that are on target. And so you'll see fewer reads aligning with the same mutations. And typically we're looking for at least 10 reads at a given loci, you know, calling a SNP in order to make that genotype call. So, you know, you start to see a number of things fall off. But you know, doublets and multiplets are generally quite easily and readily detected and removed from data.
Great. So another question, does Mission Bio have any products or future plans for analysis of cell-free nucleic acids and proteins?
That's another good question. You know, cell-free is you know, a very hot area, and there's so much work going on in the field. Right now, we are focused on the cellular context, you know, which focused on the multimodal approaches that I shared today. We don't currently have, at least prioritized on the roadmap, a cell-free approach for this technology. It's difficult because you would not really get a single cell approach by encapsulating cell-free DNA. You wouldn't know whether or not it's from the same cell or not. And so you may get potentially some sort of sensitivity improvement, and that I'm not sure of, and I certainly don't think we've done any work in that space. But with our focus on resolving, you know, clonal architectures and the single cell level, you would not be able to get that from the cell-free approach.
Great. Can you talk more about the value that the combined protein DNA multi-omic workflow provides?
Sure, yeah. So, you know, multimodal characterization really enables us to consider heterogeneity beyond just a single analyte, to understand how co-occurrence of genotypes may impact immunophenotype or clonal fitness at various stages along a differentiation cascade as defined by the immunophenotype. You know, it also enables a powerful capability to identify pathogenic clones in particular compartments, you know, throughout the cascade there. For example, in CD34, 38 stem and progenitor populations, as opposed to more mature cell types. So you can actually see where the mutations arise, which may have therapeutic implications. But the hope is that, you know, by combining these multimodal abilities and these novel characterizations of clones, it will help to inform novel strategies in the clinic and approaches to actually realizing precision medicine. The more data that we get on any individual cell, you know, the hope is we'll get that much closer to realizing, you know, actual precision medicine in the clinic.
Great. So can single cells be isolated from FFPE blocks?
The answer is yes. It's been done in the field. It's not something we currently support. A number of our customers are very interested in that, and I would posit that many people are working on it. But and it's something that's certainly on our roadmap, but it's not something that we currently support and don't have a timeline for supporting. It's obviously a challenging problem at hand, given the many different facets and considerations for single cell assays, particularly in droplets. But I do think that something will be on the horizon before long. It's just not something we can currently do ourselves or that we support on the Tapestri platform.
Great, thanks, Aaron. COVID-19 is receiving a lot of funding currently, and a lot of labs are focusing on it either in research or testing capacity. Are there any applications Tapestri can be used for the projects?
Yeah, that's another great question. You know, there's been a ton of data really showing the implications of immune status and COVID-19 outcomes, and it's such a fast-moving field that you know, it seems to change day to day. There has been some interest in using Tapestri to characterize things like inflammatory precursor conditions that may impact disease course and outcome in sort of pre-cancer and cancer models. And there's been some thought and questions as to whether or not, you know, there are ways to study those types of conditions in the context of COVID-19. But to be frank, you know, there really hasn't been a broadly defined genotype, immunophenotype question for COVID-19 on the Tapestri platform at this point. But you know, as I mentioned, things are changing. There's been a lot of discussions and interest, and certainly something we're interested in exploring. So if if there's folks on the line that have interest in discussing that, we'd certainly be all ears. But at this point, we've not largely been involved in those efforts.
Great, thanks. So it looks like we have 2 more questions before our Q&A session will be over. So does Mission Bio have plans to focus on fusions in addition to CNVs?
Yeah, so fusions obviously have a lot of clinical importance, and there's a lot of interest around this as well. What I'll say about that is right now, the way that we build custom panels would require that you have prior knowledge of fusion partners, and then we can build targeted panels that are able to detect and characterize those fusions. But we could not do that without prior knowledge of the fusion partners. There's potentially ways in the future that we could get at that, and that's something that personally I'm quite interested in and have been advocating internally for us to work on. I think that it's something that will require additional work, but we do have the ability to do it right now if you have clinically implicated and recurrently fused regions, and we can certainly incorporate those into panels.
Great, thanks. It looks like our final question of the webinar is, do you recommend separating cells with flow cytometry to reduce sequencing demands?
That's really up to the investigator leading the project. I mean, flow can be used certainly for live dead sorts, and if you're looking to run viable cells, that might be a way to clean up your sample. Generally speaking, our customers are running whole bone marrow or whole cell suspension that has not necessarily been sorted. If you really want to look with exquisite sensitivity into a particular compartment, sorted by flow cytometry, you absolutely can do that. Just keeping in mind that we do have input requirements on the genotyping side, a minimum of 100,000 cells, and so if you do sort into that, you would into a particular compartment based on an immunophenotypic marker and flow, you would need to ensure that you would have sufficient input to complete the genotyping assay on the platform. But it's not.
Thank you, Aaron.
Actually, real quick, I just wanted to add. As far as sequencing burden, with this being a targeted assay, sequencing is typically a little less of a burden from a single cell perspective than other folks that are maybe more used to whole transcriptome single cell approaches are thinking. Because it's targeted, typically we're recommending somewhere around 60 to 80x coverage per variant per cell, and you're talking about a couple 100 million reads typically for your average 200 to 300 amplicon panel with this approach. And so the cost, generally speaking, from a sequencing perspective, can be quite a bit less.
Great, thank you. Well, we've reached the conclusion of our event today. I want to again thank Aaron and Kevin for the great presentation. If you do have any additional questions, please reach out and we will make sure they get answered. We hope you all enjoyed the presentation, and on behalf of Agilent Technologies and Mission Bio, we wish you all a great day. Thank you.